So if we're starting here at the notebook, again, the goal of this section is to show you how to load meshes, and there's two different ways to do that. We'll then look at combining that with the synapse information and some of the synapse tables that Casey was previously talking about, and how to map synapses onto your mesh. And then we'll go through a couple ways to visualize your cells, both in 2D and 3D. And once you have a mesh loaded with your synapses mapped onto that mesh, you can then use graph algorithms to start doing different types of analyses. And as a prerequisite, make sure if you use the Conda environment that we had set up, you should already have Mesh Party. If not, please make sure to install that. And again, you can link to the two example meshes here. And instead of using the file with all of the synapses, we'll be using the SOMA subgraph synapses, which is just a subset. So it's a smaller file. You should already have it in your data folder. And those are all of the synapses between excitatory cells that have their somas within our layer 2-3 volume. So we'll start out by just importing a couple necessary packages. The only unique one here is Mesh Party. And then, as I mentioned, there are two ways of which you can load in a mesh. The first is if you already have your mesh pre-downloaded. So if you have downloaded those meshes off of Figshare, what you want to make sure you do is tell me where the data is. So make sure you've defined your data directory to the proper, to wherever you have it downloaded. And the second way, which I'll show in just a second, is you can directly download any mesh off of from NeuroGlancer if you have the segment ID and through using Cloud Volume. And so Cloud Volume is a package that was developed by our collaborators in Princeton, and it just allows you to read and write from the data that you see on NeuroGlancer. And Sharmi will go into how to do that in the next section. So here, the segmentation source is how we access the 3D segmentation data. I have it predefined for you. If you don't have this, you can go to the GitHub page or on your NeuroGlancer link. It is held within that JSON. And what you want to do is define this mesh meta object, which just allows you to encompass the cloud volume information. So we'll run this. This is just defining points for the data so I can access it. And now that I have already predefined my segment ID, but again, if you've chosen a cell of interest, you just want to change this so that you point to your unique object ID. And the first way I'm going to show you how to load in the mesh is right off of disk. So I want to make sure that this mesh directory is where your data is. And then you can just put in mm.mesh with your mesh file here, and that should read right off of disk. And so that's usually quite fast. If you want to make sure that you actually do have a mesh here, you can look at the number of vertices and the number of faces of this object. So these are non-zero. It means I actually have data stored. So this is the first way of which you can load in a mesh. Let's say you don't have the mesh pre-downloaded. All you need is to have the segmentation source, which I had defined up above, and your unique segmentation ID. So here I've got a second ID. And for the second mesh, rather than reading it off of disk, I'm going to download it directly from Cloud Volume by running this here. And so to make sure, again, that this worked, I have a non-zero amount of number of vertices in my second mesh. So here I have two excitatory cells loaded into my notebook. And so now I can start working with them. But let's say you want to figure out, well, where are all of the synapses onto this mesh? The first thing you need to do is read in the SOMA subgraph file. So again, this should be in your data directory. You can also use the very large synapse file if you would like to look beyond this cell of interest. So we're reading in the SOMA subgraph, and just to give another overview, as Casey went through, what this data frame now has, it has your pre-root ID, your post-root ID. It has the actual location in both nanometers and voxels. And what's unique about this table is it also has spine volume, which is not the case for the large over-encompassing synapse table. So I have all of the synapses here between all excitatory cells with somas in the volume, but I'm only interested in those that are synapsing onto my cell of interest. And so here, all I'm doing is filtering out this large soma subgraph for the postsynaptic cells on my postsynaptic synapses of the cell of interest. And this synapse XYZ is just creating an array with the XYZ positions in nanometers. And so if we run that. So now I have the mesh vertices for my cell of interest. I also have all of the synapses onto this particular cell. Let's create a 2D visualization just to look at it and make sure that it makes sense. So here I'm just using simple matplotlib scatter plot where I'll be plotting a small point for all of the X and Y positions of my, for each vertex in my mesh. And I'll do the same thing with the synapses so that I can see them here as well. So what I'm looking at here is just a very simple scatter plot, but I can clearly see this is my excitatory cell in blue, and in large purple, obnoxiously large purple dots here, you see where the synapses would lie on the cell. Okay, so if I do want to run, or if you want to run any graph algorithms on your mesh based on the synapses, you want to be able to map the synapses to your mesh. And so to do so, I'm going to filter this a little bit more by taking another subset of this. So I previously loaded in a second mesh. So if we go back to NeuroGlancer, I have these two excitatory cells, and I want to see all of the synapses between them. So what I'm doing here is I'm taking a further subset where I'm now only taking the synapses if the pre-root ID matches that second cell that I had outlined earlier. So if we run this, this has now made my synapse data frame even smaller, and it will only encompass the synapses between these two cells. And in order to map it to the mesh, I'm going to run KD-tree. And so what that means is I'm going to map each synapse to the closest mesh vertex on my cell or on my object. Okay, and so here, this will show all of the distances between the mesh and the synapses. And so you can see there's roughly 5 synapses between these two cells, and they're within 67 nanometers. That's the furthest distance. So they're quite close. And so now let's say you want to run a graph algorithm. You want to see what is the distance or the shortest path between some of these synapses. To do that, we're going to import the sparse package from Sci-Fi, which allows us to do this. And we'll run a simple Dijkstra method here. And what will that will give me is a distance matrix as well as a path matrix. And so this predecessors matrix, in other words, will give me from every point kind of what the path is to this first synapse, which I set kind of as the root here. And so for sake of an example, let's see if we can map the path between the very first synapse and the fourth synapse. And so this here will do that for me. And let's make another 2D visualization. So very simple to the one we made before, except this time I'm going to add a line to delineate that path between the first and fourth synapse between these cells. So the only thing I have added to the code from the one before is mapping the path. And so if we run this here, you can see in dark blue, there's the path between the first and the fourth synapse. And you see that I've changed the coloring. And now that I have the synapse information and where they are on the mesh, I can recolor every vertex on this cell based on this distance from the very first mesh. And so the darker the color here, the closer it is to the first mesh, which makes sense as this happened to be the first synapse here. And so the darker the color, the closer it is to the synapse. And as we get further away, you start getting into the greens and the yellows, and that means it's quite far away from that first synapse. And again, that dark blue is the path that we've been calculated on the mesh. So this is just an example, a simple example of some of the type of analyses you can run on your mesh. Some of the more exciting bits is just visualizing it. And so I'll show you a couple ways to how to visualize these cells in 3D. And so there are two packages. The first one I'll show you is called ITK widgets. This allows you to do really quick and simple visualizations within your notebook. It doesn't give you too much control over the actual visualization, but I tend to use it when I just want to make sure that what I'm doing is correct. And if I'm doing any mesh filtering, that I'm seeing the correct thing. And so here what you want to do is you need to create poly data objects. And so if you have downloaded Mesh Party, a really helpful part of that is TryMesh VTK. And TryMesh VTK has multiple functions in there that will allow you both to get your meshes in the correct type for both ITK widgets and VTK, which I will talk about in just a second. And so here what you're doing is you're converting your mesh to polydata. You're converting the path as well, so your skeleton as well into a polydata format. And then you're defining this viewer. And this viewer is going to take in these polydata objects. So here I have the polydata for my mesh. So this is the cell, and the polydata for the skeleton path. So this is still going to keep that path between the first and the fourth synapse. You can then define different colors and opacities in order to make it a little easier to see. And so if we run this, it takes a little second. Okay, and then we call on the viewer. This should pop up within your Jupyter notebook. And you can see now I have my cell of interest in yellow, and the path, if you just scroll in, as you zoom in, you can see that same path between that first and fourth synapse is in blue. And so this allows you within your Jupyter notebook to just quickly throw this in 3D and double check quality control or just make nice visualizations. So the other package that is also for 3D viewing is called VTK. This is another open source package. And this one I tend to use if I'm trying to make more complicated figures or even making a movie. It gives you a little bit more control over the camera and the types of things that you can visualize. So the only difference here is rather than a polydata object, we've defined it as an actor. So rather than a viewer with polydata objects, VTK will have a camera with actors. And so what we're doing is using TryMesh VTK again. You're going to create a mesh actor from your mesh object. I'm going to put in our second mesh so we can see both excitatory cells again. And now I'm creating a point cloud actor, which is actually going to create a 3D sphere for every synapse between these two cells. Again, you can denote the size. You can change the size. I've made these very large so that we can see them and play around with the colors if you choose. Again, so the camera object here, all it needs is what the start point is. And so I've given it the centroid of my first cell. And you can also decide how far or zoomed in you want this camera to start out. And then lastly, you want to use TryMesh VTK's render actors, which you can pass in as many or as few actors as you want. And since I have created a camera object, I'm also passing that through. So if I run this, one thing to note about VTK is that it will not run within your Jupyter notebook. So it'll come out as a pop-up, a little Python pop-up. And so here you can see my original cell. Oh, this takes a second. Let's try running it again. Okay, there we go. So here now I have a 3D view of my cell of interest here in gray, the second excitatory cell here in blue, and you can see the synapses between these two cells are these spheres. And so if I decide to zoom in on one of them, let's see this guy here, whoops. You can see that this is in fact a synapse point as the intersection of the blue axon and one of the gray spines. And so this, using VTK, allows you to make more complicated visualizations, as well as small movies and videos if you choose as well.